The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and welcome to Lab Manager's Tech Trend webinar series. I'm Tanuja Kopal, contributing editor for Lab Manager, and I will be moderating today's discussion on the challenges you encounter for sample preparation, specifically for your chromatography analysis. We will be joined today by two technical experts from Agilent Technologies and Metrome USA Incorporated, who will be talking about some commonly encountered issues with sample prep for chromatography. And they will also point out various techniques that will best serve certain types of analytes and applications. We always try to keep things very interactive and informal, and we would like you to make use of this live event to pose questions and have them answered by experts in real time. You can send along your questions or comments anytime during the webinar by typing it into the question box that appears on the right-hand side of your screen. We will address all your questions right after our speakers have gone over their brief presentations. Today's webinar will also be available for viewing on Lab Manager's website, which is www.labmanager.com, for at least 6 months. The website also hosts a number of resources such as application notes, white papers, links to past webinars, and you may find all those very useful. In our closing slide, we will also share with you the email addresses of our speakers so that you can reach out to them directly for a copy of their slides or for further guidance on this topic. You will not be able to download a copy of their slides from our website. So with that, let me turn it over to our first speaker, Dr. Joni, who is a sample prep application scientist in the Chemistries and Supplies Division at Agilent Technologies. Joni has published several application notes. She has conducted numerous seminars, videos, and classes on sample prep for customers worldwide. And she's directly involved with supporting and assisting individual customers with their sample prep needs. So I'm happy to welcome Joni to our webinar, where I'm sure she can help you with some of your questions as well. Welcome, Joni.
Thank you, Tanuja. Hello, everyone. I greatly appreciate that you are with us today, and we both hope that you find the information that we're going to provide today useful and helpful. What I'm going to talk about today is sample preparation, choices and approaches, when just enough to when just enough is not enough. I'll first start with just some intro to sample preparation for those of you who may not be using it at the present time. Then we will focus first of all on just enough techniques. That's a new terminology that's being tossed around nowadays. And that'll include things like basic filtration, protein precipitation, modifications for liquid-liquid extraction, namely supported liquid extraction, SLE, catchers, and toxi-tubes. And then we will go to when just enough is not enough, namely solid phase extraction. Now, basically, why is sample preparation important? I mean, we'd all rather not do any sample prep if we could avoid it. But many times, the sample is too dilute. A good example of this would be water. You may have to deal with hundreds of mils to liters of water to get those trace contaminants. Or it may be too dirty, as we see in food or soil, in which you've got to get the material in a form that you can then extract out the analytes of interest. The matrix may not be compatible with your analysis conditions, like possibly oil or gasoline or alcohol, or you may need to do a solvent switch. For those of you who are familiar with sample preparation, you know, and this is not unusual information, that the sample preparation process is very error-prone. It's one of the largest percentage of issues where you will have errors. And it's also the largest percentage that you will spend your time on. That's why a lot of people would rather not do sample prep. However, you have to be aware that not removing the interferences that are effectively removed by sample preparation will cause poor chromatography, mechanical issues like particulates and blockages, lifetime issues with your columns, instrument downtime, carryover. Carryover is something we don't usually think about, but interferences that lay within the system will act as active sites, causing carryover issues. Increases in sample runtime and cost, and you'll actually start to see chromatographic issues that will cause you to rerun your samples again and again. Ion suppression in mass spec is a big issue that a lot of us face nowadays, especially with how sensitive our mass specs are. And the bottom line is overall loss of sensitivity. When we think of sample prep, you have to be aware of 5 factors. 1, the analyte. What is my analyte? What is the concentration of the analyte or compounds that I need to achieve? And what is my analyte then? What's the sample matrix? What type of instrumentation do I have that I can use to analyze my compound? And the required sample size. Am I going to have enough to work with? As many of you know, there are numerous sample preparation techniques. The time allotted to us today will not allow us to touch on all of these. And as you can see here, the tools that I'm showing you are mostly for solid and liquid type samples. But what you can see starting from the bottom, going all the way up, is that the simpler, more generic methods include things like filtration, precipitation, dialysis, through to catchers, liquid-liquid, solid supported, up to the very specific ones like immunoaffinity. As you go up the types of tools, you actually achieve a greater selectivity and optimum sample cleanup. And what I hope to show you today is how you can choose based on your sample matrix. At Agilent, we have a huge variety of products that you can choose from. The ones I'm going to focus on today are Captiva. The ones on this left-hand side are what I consider the just enough type techniques. Captiva is designed for protein and lipid remover filtration. Chemolute will actually mimic liquid-liquid extraction. Catcher's Bondilute, which will allow you to analyze contaminants from food and other types of matrix. It's one of the only types of sample prep that you can actually use a solid. And last but not least, the more specific and the cleanest through solid phase extraction, namely the silica and the polymeric SPE. Now, as I mentioned, I can't touch on everything, but what we're going to look at, first of all, is to start with dilute and shoot, because I know a lot of you are familiar with that. We will then move into filtration, namely the Captiva and the syringe filters, and then into our first strategy, which is protein precipitation, which is inexpensive, works very well with biological samples, and can remove both proteins and lipids. Then into liquid-liquid extraction, which is our second strategy, namely SLE, supported liquid extraction, catchers, and toxitubes. And last but not least, our specific and cleanest when just enough is not enough, solid phase extraction. As I mentioned, a lot of you are familiar with dilute and shoot. Many people will say, all you need to do nowadays with these mass specs, because they're so sensitive, so selective, just dilute your sample. I'm here to tell you that that is not a sample prep technique. All you're doing is diluting your sample, which means the interferences are still there. They are not removed. Yes, your column may last a little longer, but you are still going to have a problem sooner or later. And that is an issue that we all need to be aware of. We do need to do some type of sample preparation. Many times, one of the issues that we have with sample preparation is just making our serial dilutions, our extractions, or standard additions. We have an automated instrument called the workbench. This will actually automate your dilutions, your aliquoting, and your reconstitutions offline, so that takes the manual steps out of the procedure and actually increases and optimizes your precision and accuracy. Why filter a sample? This is the simplest type of sample preparation technique that you can do, and it is very, very important, especially if you are doing UHPLC. Why? Because with UHPLC, you are using sub-2 micron particles, which means the frit is a 0.5 micron, which means it will clog very, very easily. By filtering the sample, you reduce that issue, and in fact, with UHPLC, you ought to filter your solvent. So if you can't do anything, you should at least filter your sample. Our first strategy is specific for biological that contains proteins and lipids, and that would be considered protein precipitation. It's a very low cost and low complexity, and also gives you quite clean samples. It's usually used in conjunction with LC-MS analysis. How you do this for the protein precipitation can occur in many ways. Addition of salts, low pH, metals, and in our case here we're going to talk about organic solvent. Usually occurs with a 1:3 ratio. So in other words, 1 part of your biological sample to 3 parts organic. Then you either follow by centrifugation. Here's an example of adding the organic to a plasma sample, or by filtration, namely with our Captiva ND. ND stands for non-drip. As I mentioned, the protein precipitation is a just enough technique, so will not be as clean as other methods like SPE. Implementing the Captiva ND protein precipitation plate actually decreases your time for protein precipitation by 5 to 6-fold, because you can do the entire protein precipitation in the individual wells. No transfer or centrifugation is required. So there is no chance of loss of sample. So what does that equal? That equals a huge cost savings for your lab. The use of the 96 well or the cartridge formats for the Captiva ND and ND lipid will facilitate automation. It's very easy to use. There's no cross-contamination or sample loss from dripping. As you can see here, there's actually acetonitrile sitting in these wells, and no amount of acetonitrile is dripping through the tip, which can be a major problem with protein precipitation plates. Better reproducibility than what you will get with centrifugation and manual protein precipitation, and it's greater than 99% protein removal as shown by the Bradford assay. Here's an example of using the Captiva ND plates with a UHPLC column. After 5,000 injections, we don't see an increase in pressure, which correlates to no blockage of the frits. So therefore, implementing something as simple as the Captiva ND plate or column will drastically increase the amount of injections you can get onto your UHPLC column. One of the issues, though, with protein precipitation is that lipids, especially in human samples, are still soluble. And having lipids there will influence ion suppression or data reliability and your sample downtime, and that's something we need to realize. A way of approaching this is to actually use advanced precipitation technology, which uses the same idea where you add your organic and your biological sample to the well, mix it, and as you pull a vacuum, the material not only precipitates the protein, but depletes the lipid from the sample. So therefore, you reduce the lipid content. This is actually a chromatogram showing that removal. The green is just using protein precipitation. We still have a substantial amount of lipids in there. But with the Captiva ND lipid plate, we have removed over 95% of the lipids, shown here by the red chromatogram transition from 184 to 184, the normal transition for monitoring of phospholipids. Our second strategy is based on liquid-liquid. It will incorporate Chemolute, which is a solid-supported liquid extraction, catchers, and toxi-tubes. For those of you who are not familiar with liquid-liquid extraction, it involves mixing 2 immiscible solvents. When you do that, you hope that your analytes of interest will go into the organic, which you can then separate out, reduce down, and analyze. It usually involves a lot of volume. You usually have to use a SEP funnel, maybe even chlorinated solvent. So it's not a very green technology. It's also not selective, and it is extremely difficult to automate. A new approach using sorbent supported liquid extraction, or SLE, actually mimics liquid-liquid extraction without all of the problems associated with liquid-liquid extraction. You use a cartridge that it has diatomaceous earth in it. There is no conditioning required for this cartridge. All you need to do is apply your aqueous sample, allow it to sit on the diatomaceous earth for about 5 minutes, and then come back with your organic solvent, collect, try it in 2 shots. That actually gives you a better recovery across the board. What this does then is mimics the liquid-liquid extraction for you without all of the problems. Supported liquid extraction applications are very wide-ranging, anywhere from azo dyes in textiles, caffeine, pesticides in honey, polyphenols in wine, acrylamides in food, phthalates in cosmetics, and whiteners in cosmetics. The solid supported liquid-liquid extraction has many benefits over liquid-liquid. First of all, no emulsions, no special glassware, less time, minimal method development. Does require an aqueous solution though, and that's something to keep in mind. It is a just enough technique, just like we saw with protein precipitation. It reduces technique dependency. You will not see variances between different people doing it or different labs, increases reproducibility, and can be automated. Our third type of just enough technique is called catchers. Catchers stands for quick, easy, cheap, effective, rugged, and safe. It allows for the simultaneous determination of many classes of compounds at once. It was developed for pesticide analysis from food. It is a 3-step process, namely extraction, dispersive SPE, and analysis. It will allow you to analyze contaminants, and it's usually based on established levels, like considered the MRLs, or maximum residue limit. The levels needed to be attained are based on these MRLs, or limits that have been determined to deem terrible or cancerous or issues with human or animal life. The lowest levels of detection are usually not required for food safety because they set these MRLs. And the reason why catchers is an available just enough technology is because of the sensitivity and selectivity that we do have now available via MS and tandem MS. As I mentioned, catchers was designed for pesticide analysis. However, it's not just for pesticides anymore. PAHs have been done, PCBs, illegal dyes, mycotoxins, veterinary drugs, antibiotics, pharmaceuticals, acrylamides, and many more. There are over 1,000 applications articles per year done on catchers, and it was just developed back in 2003. The procedure is to take a sample. It can be solid or a liquid, homogenize it, add it to a tube, add solvent, shake, add your salt, add internal standard if required, shake, centrifuge. You will end up with a tube with these separate layers. Take an aliquot of the top layer, add it to a dispersive tube containing SPE material, vortex, centrifuge, you're ready to analyze. You take an aliquot of the top, you can go to GC or LC analysis. Catchers over liquid-liquid, catchers methodology is as easy as 1, 2, 3. It's a just enough sample preparation technique. It does not require the sample to be a liquid, which is unique. It takes minimal sample preparation technique, expertise. If you can pipette and you can shake, you can do catchers. It gives excellent results. It's a very green and cost-effective technique, and the capabilities are even surprising the experts. The last type of just enough technology that I'd like to talk about is toxi tubes. You will see that it is very much like catchers, but this is designed specifically for the clinical and forensic drug analysis. It involves adding the sample, mixing, and centrifuging. It is designed for challenging matrices like postmortem blood, gastric content, and liver tissue. There is absolutely no method development required. What you need to do, and as I showed you, this is similar to what we saw with the catchers, you will add your sample, mix, and centrifuge, as shown here to the left. You will then take an aliquot of this top organic layer, dry it down, and then reconstitute for either GC or LC analysis. Our last but not least is when just enough is not enough. And we're going to talk about solid phase extraction. Solid phase extraction will give you the ultimate cleanest sample. It eliminates all of your interferences. It is customizable, and we have simplified methods for ease of use. The basic steps are to load your sample, wash it, and elute. It is the best at removing interferences. You gain a concentration step, which is a unique feature of solid phase extraction. It is very flexible, highly automatable, and it can extract a whole suite of drugs. Many people feel that it requires a lot of method development and additional work is involved, but I'm here to tell you that's not the case. There are 4 basic chemistry mechanisms associated with solid phase extraction: hydrophobic, polar, electrostatic, or mixed mode. Mixed mode is available in many of the polymeric materials. Polymeric SPE has many advantages over basic silica. First of all, it's stable over a wide pH range, so it is useful for all methods. It is resistant to drying, which can be a major issue that people face using SPE. So you don't have to worry about that decreasing error with solid phase extraction. It has a very high capacity, 5 times that of silica. So you can use a smaller bed mass, minimum solvent usage, and greater throughput. And last but not least, which is one of the basic keys, it has simple generic methodologies associated with it. So that we have great places that you can start. You don't have to reinvent the wheel. SPE applications range across the board: pharmaceutical, biotechnology, environmental, forensic, food safety. Everything can be done essentially with solid phase extraction. This essentially is a table that I hope you use either now or in the future. What it is, going from left to right, shows you from just enough to when just enough is not enough, and the types of interferences that each of these techniques remove, from particulates down to polar organics, and the possible formats that are available. So I won't go into detail here, but I hope you use this when you're trying to choose a type of sample preparation technology. In the future, if you would like to go to our website, www.agilent.com/chem, we can address any of your sample preparation techniques. If you go to our website, a window like Step 1 will pop open. Clicking on Columns and Supplies, you will access sample preparation. Step 2, or this window, will become active, where you can access our selection guides, shown here. You can even download them to your iPad so you can keep them for when you're at the beach this summer and you need something to do, or our catalog. And we also have numerous applications defined by sample preparation apps, SPE apps, or catchers application. If you'd like to review in more detail any of these techniques, you can go to our education and events, and you can take a look at our SPE videos and our 2 catchers videos. The intro to catchers, obviously, is the introduction to catchers, and the catchers number 2 is for best practices, and you can review them now or in the future, or download them for your researchers to take a look at. That concludes my talk, and I greatly appreciate your time.
Okay, thank you, Joni.
Thank you.
And thanks for all the questions that have already come in. For those of you who may have joined us late, you can submit your questions or comments anytime by typing it into the question box on the right-hand side of your screen. And also a reminder that the webinar will be available as an archive on Lab Manager's website, and all of you will receive a link to that archive probably next week. The email addresses of our speakers will also be shown to you in our closing slides. So please feel free to reach out to them directly if you're looking for a copy of the slides. You will not be able to download any slides off of our website. So our second and last speaker for today is Dr. Harina Hymen, who is the Technical Manager for Ion Chromatography at Metron USA. Hari is the company's go-to applications expert and runs their application support department. He has spent nearly 15 years working with a wide range of techniques and applications for elemental analysis. Hari specializes in sample prep techniques for ion chromatography, and Hari was a part of our elemental analysis webinar last year, and so it's a pleasure to welcome you back this time.
Thank you, Dhanuja. And so far we were listening about the organic molecule applications and the related sample preparation from Joni. And I'm going to talk about the latest in sample preparation technique that is to do with the ion analysis. So as we know that the ion chromatography, the part of the classification of an HPLC, has grown and has got developed as a very important analytical technique for the trace level ion analysis. There are a lot of development as took place in terms of the miniaturization of the instrumentation, starting from the several intelligent components like intelligent pump, intelligent column, intelligent detector. Everything is put together. And also the column dimensions, there is a greater amount of development from a 4 mm column to a 2 mm column. And all this development on the instrumentation side has really expanded the applications of IC as an analytical technique, which is being widely used for the anions, oxyhalides, and the organic acids applications, alkali and the alkaline earth metal applications, and it offers the nanogram per liter detection limits based on the development in the instrumentation. But the key point is that the challenge remains with the sample matrix and the associated sample preparation. How do we integrate the needed sample preparation to the already developed ion chromatography technique? From my point of view, I would like to classify the sample matrix, especially related to the ion analysis, into the 4 different categories. A sample matrix which has got no interference or no damage, meaning that we are talking about the pure standards here. The sample matrix that may have an interference on the chromatography, but it does not interfere with the column or the suppressor. The sample matrix which could really damage either the suppressor or the analytical column, but need not necessarily interfere with the chromatography system. And sample matrix which can really damage or as well as interfere with the measurement, which is an ideal technique for the different type of sample. An ideal sample should be like free of particles and no interfering matrix. Concentration, it's in the ppm range, it's ready for the direct injection, and it's there in the aqueous solution, and the pH is right about 7. So we see this ideal sample only in the laboratory where we prepare the aqueous standards. The true real world samples will have a lot of particles, protein substances. The matrix may be really harmful to the column. It could be like a very high concentration or very low concentration of the analyte of our interest, emulsions, and the samples can also have the extreme pH of 0 to 14. When we talk about the associated sample preparation, depending on the type of samples, which contains high amount of particles, we need to employ the ultrafiltration. The high analyte concentration really requires the appropriate sample dilution. The interfering cations like the transition metals needs to be removed before introduction into the IC system. And if it is an insoluble component, it requires appropriate liquid-liquid extraction. The dialysis for the low concentration of the analytes, the preconcentration, and the matrix elimination, as well as the neutralization techniques. What happens if we do not do this appropriate sample preparation? One aspect is the interference. The interference could be the coelution, and some of the species could be highly retained, and they can really elongate the total runtime. And it can also lead to very low recovery, and we can also see in terms of an unstable baseline. These substances can also interfere in the measurements, like in terms of the damages. Either they can damage the column, which could be due to the particulate matters, the organic substances, high molecular weight substances, as well as the heavy metals. The suppressor, which is a key component in the IC, can be very easily damaged by the presence of heavy metals, as well as the high molecular weight organic substances. The associated sample preparation, either we have got two choices. We can go by the traditional manual sample preparation, which is tedious and the time consuming, or we can automate the entire sample preparation by the inline sample preparation methodologies. When we talk about the automated sample preparation techniques for the sample matrix that contains a high amount of particulate matters, it's important that the sample needs to be filtered using 0.45 micron or less than that pore size before it's introduced into the IC system. Because the particulate can really clog the injection valve, and the bacteria that may be present in the wastewater sample matrix can grow and can foul the IC column. The inline ultrafiltration system, which is dedicated and it is suitable for the anions and the cation analysis in the ppb to the ppm range, and best suitable and recommended sample preparation technique, especially for the wastewater, processed water, and the surface water, including the drinking water matrix. Here we are showing the typical setup for the inline ultrafiltration setup. The sample is transported from the sample processor to the right-hand side onto the IC injector via the ultrafiltration cell. Here we are making use of the dual channel peristaltic pump. The first channel transfers the sample into the ultrafiltration cell, and the second channel of the peristaltic pump transfers the filtrate to the IC injector. How does it work? So we have the high sample flow rate at the ultrafiltration cell on the sample side. The sample goes and then they get filtered, and the filtrate is aspirated by the second channel of the peristaltic pump, and it gets transferred into the IC injector. Since the sample flow rate is relatively higher on the bottom of the ultrafiltration cell side, the particulate matter that may be staying onto the ultrafiltration membrane is continuously getting flushed out, and hence there is no clogging of the membrane, and one ultrafiltration membrane can be continuously used for several hundreds of samples. The example, the anions in the wastewater, like the paper and pulp plant water matrices, is that here the sample is diluted 1 to 50 and then passed through the ultrafiltration cell membrane. Second, we are talking about the high concentration or the high salt content in the different sample matrices. The high analyte concentration can really overload the analytical column, and sometimes it may not be possible to have the good baseline to baseline separation of the analytes of our interest. So it's really recommended to do an appropriate sample dilution. The inline intelligent dilution is then a recommended sample preparation technique, which is best suitable for the ppm level to the grams per liters of the analyte concentration. Here the intelligent dilution can be performed into the external vessel or inside an IC sample vial itself. And it is the best suitable technique for the drinking water, groundwater, as well as the processed water and the wastewater sample matrix. We know that traditionally we perform the manual dilutions in the laboratory to bring down the concentration of the sample matrix. And we also know the associated disadvantages of manual dilution, like the error-prone, time consumption, and they are material intensive. Now, with the latest development in the intelligence built into the IC system, and also the enormous amount of intelligence built into the IC software, it's possible to perform a logical dilution automated. In this case, the user simply starts the determination, and while the injection is done and the chromatography is going on, the system continuously checks the area of the analytes of our interest. If the area of the sample is smaller than the highest standard, meaning that it is well within the established calibration curve, then the system moves on to the next sample. In case the area of the sample is bigger than the highest standard of the calibration, meaning it is away from the established calibration, then the system can automatically re-inject the sample to fit that into the established calibration curve. The entire process is completely automated, and from the analytical chemist point of view, you can just leave the sample. As I explained it earlier, the logical dilution works on the basis of comparison of the highest standard area versus the sample. If the highest standard area and the sample ratio is calculated, and based on that, the system can really decide whether a dilution is necessary or not. Using this system, it really takes out a lot of the manual sample preparation, and this really ensures that there are no more results outside the established calibration curve. That means no more manual re-dilution and the associated recalibration due to the re-injection of the manually prepared samples. The logical dilution setup also allows the user to perform the automated calibration from the single stock standard. So in this case, we prepare like one stock standards of the anions or the cations, and the system can perform the dilution and then establish the calibration curve. For the known samples, we can also do the direct injection, and for the known matrix where you need an approximate dilution, the system can also perform like the fixed dilution, or it's also capable of performing the intelligent or the logical dilution. The setup for the logical dilution looks like this. On the right hand side, what you see is a standard anion IC system, and on the left hand side, what you see is an automation with the external dilution setup, and in the middle of the instrumentation, what you see is a liquid handling device called as a dosino, and it has got 1/10,000 of the resolution, meaning that depending on the burette volume connected to the system, from few microliters to the fraction of a microliter can be pipetted out using this device. So the first step is that the known and precise volume of the sample is aspirated by the liquid handling device dosino. Then the arm is moved into the external dilution vessel, and the pipetted sample together with the diluent, in this case it's an ultra-pure water, are transferred into the external dilution vessel. It gets mixed completely, and then the diluted sample is transferred into the IC injector. It is also possible to carry out the dilution instead of the external vessel. It's also possible to do it in the IC vial. So the nice feature of this setup is that it can also be expanded with the ultrafiltration together, meaning that the sample can be diluted and filtered and can be injected into the IC system. To give you an idea, the capability of the automated intelligent dilution system, for example, the 1000 ppm of the chloride is placed into the IC sample processor vial, and the system does an automated dilution of 1:10 to 1:200 to establish the calibration curve from 5 ppm to 100 ppm of chloride. So this really gives us a flexibility, like absolutely very low RFD and the very long linear calibration range with a good correlation coefficient and then RFD less than 1%. Few examples. Cations in the infusion solution, you can look at it, the potassium concentration and the sodium concentrations are in the thousands of ppm. Here the sample is diluted like 1:10 and then injected to the cation IC system. Inline dilution for the pharmaceutical products containing MDEA as far as the piperazine, the sample is diluted 1:20 here. And the anions in the wastewater of a flue gas desulfurization plant, requires an additional dilution of 1:100 followed by the ultrafiltration to remove the particulate matter in the wastewater matrix. The third type of sample matrix which is challenging for the ion analysis is the emulsions and the proteins containing the hydrophobic sample components which may remain on the column and can eventually foul the active sites available in the anion as well as the cation column. So removal of these interfering hydrophobic components is really necessary, and it is done based on the dialysis techniques which is suitable for the ppb to the ppm range of concentration, and it is mainly employed for the different strong matrices like emulsions, cutting oils, oil droplets, fermenter solutions, dairy products, body fluids like the blood, urine as well as the sweat samples, the tobacco samples as well as the fruit and vegetable juices. The basis of this dialysis setup is based on the osmosis principle, and here we are using the dialysis membrane based on the nylon or the cellulose acetate membrane, and it can be done using the dual channel peristaltic pump. On one side of the membrane, the sample containing the interfering matrices and the analyte of our interest is passed on continuously. On the other side of the membrane, we have the acceptor solution, which is an ultra pure water. This is based on the stop flow technique, meaning that the acceptor solution is stopped for the dialysis duration of 4 to 10 minutes. During this dialysis time, due to the osmosis principle, the concentration on the sample side and the acceptor side will be same after about 4 to 10 minutes. And then the dialyzed sample, which is free of the interfering matrix, is transferred to the IC injector. The typical examples are like the trace level anions like iodide, thiocyanate in the milk matrix. Here the sample is diluted and then the dialysis is performed using the 0.2 micron cellulose acetate. The cations like the sodium, potassium, calcium, magnesium in the orange juice is done also using the dialysis and sample preparation technique. The cations present in the automobile process water, here we can quantify including the cations like the zinc and the iron 2. Here the sample is diluted like 1 to 250 times and followed by the dialysis using the cellulose acetate membrane. When we talk about the low level concentration analysis, ultra trace analysis in the ppb or the ppt level analysis, it requires the sample pre-concentration. In this case, the large injection volume may lead to the peak broadening, and the broad water tips would influence the fluoride quantification. The inline preconcentration is suitable for the ppb to the ppt level analysis, and using the liquid handling devices, we can do the variable volume pre-concentration from 4 microliter to 4 ml of the sample volume. And this allows the user to perform an automated calibration from the single stock standard. And it's highly recommended for the ultra pure water analysis and the power plant water matrices like boiler feed water, condenser water, as well as the polisher water. Here, the loop is replaced by the pre-concentration column, and the sample containing trace amount of anions or cations are pre-concentrated on the respective pre-concentration column, and then they are decoupled by the element. The setup is modified here a little bit different, and the loop is replaced with the pre-concentration column, and the liquid handling device is also modified here. The modification really allows us to perform an automated calibration curve from the single stock standard. Especially when we talk about the PPT level analysis, it's very difficult to prepare the standards at this low concentration because they are highly unstable. So thanks to this flexibility in the variable volume pre-concentration, we can start with the PPB level standard, and the system can perform an automated calibration curve, for example, from 10 ppb to the 10 ppt. As an example, the lithium calibration is shown here. Here the variable volume like 4 ml to 4 microliter is pre-concentrated on the cation pre-concentration column, and that gives us an excellent linear calibration curve at this low concentration. Sometimes we may have to do the trace level of anions or the cations in the interfering as well as the harmful matrices. So here we may have to combine the two different techniques, that is the pre-concentration as well as the matrix elimination. Here the samples are pre-concentrated on the respective pre-concentration column, and the interfering matrix is selectively removed from the pre-concentration column. And this is best suitable for the matrices like the fuels and the samples with the high concentration of organic solvents, samples like hydrogen peroxide as well as the borated matrix. Here, after pre-concentration, the interfering sample matrix is washed with the ultra-pure water or with the suitable solvent. So the interfering matrix is completely removed from the pre-concentration column, and then subsequently the trapped anion or the cation in the respective pre-concentration column is transferred into the analytical column by the mobile phase. So it's like a three-step. First we do the pre-concentration, second we remove the matrix, then we transfer the pre-concentrated analyte onto the analytical column. The last but the very important sample preparation technique is the combination of inline neutralization, pre-concentration as well as the matrix elimination. When we are talking about a very harsh sample matrix like electroplating samples, Bayer liquor or caustic or TMAOH, we cannot do the direct pre-concentration because in the electroplating samples, the heavy metals present in the sample matrix would interfere. For the Bayer liquor, caustic, and the TMAOH, these matrix itself will act as an element and that will not allow the pre-concentration of the anion. And that's also applicable to the nuclear power plant borated water matrix, which contains lithium hydroxide. So in this case, before doing the sample pre-concentration, the sample is passed through the inline neutralization chamber, which is nothing but a cation removal device, which removes the sodium or the heavy metals present in the sample matrix, and then subsequently the samples are pre-concentrated and the matrix are removed. The typical example is the trace level anion application in the PWR matrix in the nuclear power plant, which contains like 3 grams of an boron as well as the 6 ppm of lithium hydroxide. The lithium hydroxide removal is very critical to do the accurate pre-concentration of the trace level of anions, and the accumulated boron needs to be removed before the sample is transferred into the IC analytical column. In summary, we need to choose the appropriate sample preparation techniques, which is mainly based on what type of sample matrix we are talking about, what is the concentration of the matrix analytes, and what is the analyte ions we are talking about, whether it's a cation or the anion, what is their concentration, and what is the detection techniques we are employing for the analysis. Is it a suppressed or non-suppressed conductivity detection? Is it the UV visible detection or the pulse amperometry detection or the mass detection? So based on these 3 important parameters, we need to choose and employ the appropriate sample preparation technique. Thank you very much.
Thank you, Hari. And as you all can see, the email addresses of our speakers are now on your screen, and feel free to get in touch with them for more information on this topic, or if you need a copy of their slides. So with that, let us get to some of the questions that have already come in. The first question is, how do you stabilize tricyclics? For example, something like nortriptyline. Joni or Hari, any advice there?
I think Joni.
So, I'm sorry, say that again for me, please.
How do you stabilize tricyclics? And the example here is nortriptyline.
Right. I'm assuming what you're referring to is out of biological samples.
I guess there's not much information provided, yeah.
Yeah, well, that's okay. One of the ways that you need to be aware of is what exactly the pH is when you're starting to work with the tricyclics, for instance, with solid phase extraction. So be aware of their pKa. Make sure that you're well outside of that range, essentially 2 units, and you can use the polymeric material, for instance, the ion exchange, to extract them, and then you can stabilize it by changing the pH back again.
Okay, and a clarification was provided that nortriptyline is a tricyclic antidepressant drug. So,
right.
Yep.
Yeah.
All right, but yeah, that's good advice. Hari, anything to add to that?
Not really from the IC side, Tanuja.
Okay, yep, got it. All right, the next question is, I'm interested in analyzing the lipids present in algae, and I see that the lipids are contaminated with chlorophyll upon the extraction. So could you suggest a sample prep technique that can separate the chlorophyll from the lipids?
I can. Graphitized carbon black.
Okay. And that should work fine?
Yeah, it depends on quite possibly a little bit of the extraction procedure before that, but graphitized carbon black is very well known to remove chlorophyll from material. The only thing that you're going to need to do is you will need to pull the lipids through with organic, because the graphitized carbon black is essentially non-selective, but the lipids should come through without a problem.
Okay, thanks. All right. Next one. If I have a sample of suture made out of a biodegradable polymer, then which preparation technique would you recommend in order to perform a successful chromatography? And I think the person is thinking in terms of GPC, so gel permeation chromatography. Would that be the ideal choice?
If the polymers of a known molecular weight or the components of breaking down of the polymer are of distinct molecular weights, then GPC would quite possibly be a good choice. There's also I think GPC would probably be the best choice, and then you could fractionate at that point in time.
Okay, okay. And also, is GPC the right choice if they have to run several different, several samples at the same time?
Several samples at this. Well, with the GPC column, sure, you can run one after. Well, you can. I think that's what they're referring to. You can run one right after another. You can collect your fractions, and if that person wants to email me, I will actually get them directly linked up with our GPC group.
Okay, okay, that's great. And here's a little more clarification on that. She mentions that the polymer will be exposed to gamma radiation, and thus I'm trying to actually quantify the radiation due to the different doses. So I guess it's a very a complex protocol, and we will be happy to pass along their contact information to you as well.
Yeah, please do. Please give me a jingle or email me, and I will do my best to address it.
Okay, great. I appreciate that. Thanks. So when we did a survey, actually, this was a few months ago, like with every other technique, our survey showed that people, especially with sample prep, they're looking for faster time, low cost, and of course more accuracy. My first question is, in your experiences, are you seeing something different other than these three most commonly asked for, things like faster time, less cost, and more accuracy? Are you seeing anything more different? And if not, then I would like to go into some details about how you can actually go about, doing that.
Harry, did you want to respond first, or I can?
Yeah, sure. For example, all the 3 points that you told, the faster, more accurate, and also the kind of a less cost of ownership. The one example that I discussed in detail, for example, the intelligent dilution in combination with an ultrafiltration could be a very nice example. For example, the environmental laboratory who are expected to test the drinking water as per the EP 300 or the different regulatory methods, it's very difficult for them to have a good idea whether the sample concentration will be within the established calibration curve or not, because they are receiving multiple types of samples from the multiple sample streams. The intelligent dilution really helps them to do an automated dilution, and that really ensures that they don't have to come back in the morning and do the manual dilution of the sample. And when they do the manual dilution of the sample, they also need to rerun their calibration. That means a lot of time goes on the doing recalibration and the reanalysis of the time. The intelligent dilution completely takes out all these requirements, and the user simply plays the sample and the system takes care of the automated dilution. And also, as per the EPA requirement, like after every 10 samples, as per the quality assurance requirement, they need to analyze the CCB as well as the CCB as a requirement. And the system also does like a counter. And then after every 10 sample analysis, including the intelligent dilution, the system injects the CCB as well as the CCB, verifying and ensuring that the collected data passes the quality assurance and they can release these numbers. Coming to the ultrafiltration, traditionally people take the a manual ultrafiltration setup, syringe type setup. Like per sample, they have to use one syringe as well as the one filter, which would be costing them approximately about $1.50. And with the ultrafiltration cell setup, they can go add and perform few hundreds of the samples, at least between 200 to 300 samples, depending on the sample type. So the ultrafiltration really brings the cost of the cost per analysis, and the intelligent dilution brings them the accurate, reliable results, and also it saves a lot of time for them.
And I agree with you, Tanuja and Harry. Yes, definitely. Everybody's looking for faster time with sample prep, less cost, and more accuracy. And that's one of the reasons why I mentioned and explained some of the just enough techniques, because they require minimal sample prep expertise. Everybody would love to avoid sample prep if they could. They'd rather give a kidney than do sample prep. And that's an issue, and everybody feels the same way. And the just enough techniques do actually give you a faster turnaround, less cost per analysis, and quite a lot of accuracy that is involved, especially with the sensitivity and selectivity that we have with our mass specs nowadays.
Right. But coming back to accuracy, I mean, is there a way for users to track if the sample prep technique itself is degrading the sample or modifying it in some way?
Well, in my case, in many cases, what you should be doing is looking at internal standard. You should, in most cases, you should be adding an internal standard. In the best scenario, it's usually the deuterated version of the analytes you're looking at, but we can't all afford that because that would be astronomical. But so you want to pick something that is similar to. By doing internal standard, you can actually monitor the extraction technique. And then also, too, in the just enough techniques, you're going to have to do matrix match calibration curves because there is still some matrix in there. And those are things that you do need to realize and take into account.
Correct. Yeah, that was the next point, about choosing the right internal standard. So I'm glad, because this is, again, I guess, a discussion on best practices. There's some things that people know but don't always do. Hari, your experiences with using internal standards?
From the ion analysis standpoint of view, this is the external standard which is commonly practiced. And here there are two ways the system performance as well as the accuracy of the results is verified. One is by, as I mentioned, those who are following the regulatory methodologies. After every 10 samples, one check standard or the verification standard is injected, and that variation is also monitored. It is within plus minus 5% or plus minus 10% variation is followed. And when we talk about the trace level analysis in the complex matrices, the system is configured and designed in such a way that it can also do an automated spike and recovery study. So it can go to the sample, it can pick up the sample, it can also spike the known volume of the standard to the sample, and we can calculate the spike and recovery, and that thereby we can validate and we can verify the accuracy of the numbers that we are getting from the complex sample.
Right. Sorry, Joni, did you have anything to add? I thought I just switched it over to Hari, but maybe you had a comment there.
No.
Oh,
we're fine. Thank you very much, though, for asking.
Okay. Hari, I had a follow-up on the automation techniques that you described. When do you see automation as a growing trend in sample prep, first of all? And if it is, then is there anything that people should consider when looking to automate? I mean, is there a certain throughput threshold, or is it very sample and application dependent? What are your thoughts on that?
It's pretty much the automation is like growing. Like as I mentioned in the beginning of the talk, one aspect of development is on the instrumentation side, the miniaturization of the column technology, and the further development on the suppressor technology. And the parallel development also has happened with the Metron, that is on the automated sample preparation, which is the key strength. And because when the people are talking about the productivity, it is not just about the sample analysis time, and it is also associated with the sample preparation time. By combining the sample preparation inline together with the IC, we are able to cut down the sample preparation time quite a lot. So the intelligent software allows us to prepare the next sample while the first sample analysis is going on. So practically, the user would see only the time taken for the analysis. The sample preparation for the second sample is going on in the background while the first sample data collection is going on. And this trend is growing, and it is continuing. And personally, I would say that more and more miniaturization technology is coming for the sample preparation in combination with the IC. That's what would really benefit the users. Because not just miniaturizing the IC is going to benefit the customer, but the user. The miniaturization and the nice tandem connections of the sample preparation together with the IC would really benefit the users world.
But what are some of the disadvantages to automating? Oh,
personally, I do not see any disadvantage, except that probably maybe people may lose some job loss, could be the one of the things. But I think, in my opinion, that may not be the big factor, because as the more and more automation comes into picture, the people can focus more on more and more creative work rather than doing the routine works.
All right. But I guess you are increasing the cost of analysis at least upfront because of all the investments you have to make in the automated tools, I guess.
Yeah, but it depends on overall, because the automated system can literally work 24/7.
Correct. Yeah.
And so it does also have a log file, so it can you could very easily monitor what's going on and where, if in fact an issue arose, which is very hard to do manually.
True.
Yeah, especially for the trace analysis, when we are talking about like PPB or the PPB level, when it comes to the IC applications, the manual sample preparation is absolutely not recommended, because you really never know from where the fluoride impurity is coming or the sodium impurities are coming up. So the best practice for the trace analysis using the IC is to use automated sample preparation to automate the calibration, also using the single stock standard.
And I mean, because we're talking about automation where we are actually eliminating all possible variability due to user, which is user dependent. But going back to manual techniques, how prevalent are the variabilities arising from the user, and what advice do you have for minimizing those?
Well, in the case of either the just enough technique or in something that's more like solid phase extraction, I think it's very important to have, when a method has been validated, is to have a written SOP that can easily be transferred within a lab and between labs. And so if you choose to manually do something, documenting everything is very important, because different techniques between people can be hard to identify if it's not documented.
Yeah. Sorry. Well, that brings us to an end in terms of all the issues that were raised. If you think of any other questions, or if you have any other issues that you would like to discuss, please feel free to reach out to our speakers directly, and I'm sure they will be happy to take some time out of their schedule to help you out. On behalf of Lab Manager, I would like to thank both Agilent Technologies and Metrum USA for their participation and support of this event. My sincere thanks to both our speakers, Joni and Hari, for putting in the time and effort to make their presentations very relevant and very informative to the users. The webinar, as I mentioned earlier, will be archived on Lab Manager's website for 6 months. So please do visit us to review this webinar, to check out past webinars, or to get more information about upcoming webinars. And I hope you can join us again next week, where we will be discussing innovations in incubators for cell culture. So thank you all once again for your time and attention. Until next time, goodbye and good luck.